Welcome, everybody. This poster is about a possible new method of imaging cholangiocarcinoma using internal coils, designed to help improve planning of resection surgery. And the authors, you can see there's quite a lot of us. We're an international team, partly based in Thailand at Khon Kaen University Hospital, partly in London at St. Mary's Hospital, partly at Philips, the scanner manufacturer, and partly in the electrical engineering department of Imperial. My name is Richard Syms. So before we start, we need to know a little bit about how MRI works. Images are obtained by having the patient lie inside a powerful magnet, inside a tunnel, and exciting the tissue in their body, particularly the protons, with a radiofrequency pulse, and then listening to the decaying echo obtained. Now, different tissues produce signals that decay at different rates. I've shown this using the graph on the left. We can see two curves, one blue, one green, relaxing at different rates. And essentially what the system does, what the scanner does, is measure these signals at a particular time and use the differences in their amplitude to build up a picture. We can see here, if we sample at a short time, the blue signal is larger than the green signal. They're both quite high, but actually we don't see that much difference between the signals. If we sample later, that's at the right-hand line, we see much more difference between the signal amplitudes, but they're both smaller. In fact, they can be small compared with the noise that's also acquired at the same time as the signal. Now, these differences in signal amplitude are used to provide contrast in the image, and they're used to allow you to tell different tissues apart. And unfortunately, you can see from this basic argument that there's an inherent contradiction between image contrast and noise. That's a key problem, especially for external coils, which are normally used in the scanner. They wrap around the human body. They pick up noise from the whole body. Typically, they give uniform sensitivity, but not very good signal-to-noise ratio. Now, the answer to this may be provided by internal receivers, that is, receivers that are inserted inside the human body on some kind of medical instrument. They have a very non-uniform sensitivity variation that's shown by the central colored map. They detect very strongly near to the coil structure itself, but the signals decay away roughly as 1 over the distance squared. What that means, if we look at the plot on the right, is you can usually obtain a signal advantage, or more specifically, a signal-to-noise ratio advantage near to an internal coil compared with an external coil. If you can get that advantage, you can then use smaller voxels to build up the image and increase the image resolution and see more detail in it. So that's the basic idea, but we have to take one more step. Because the coils themselves are non-uniform, we need to try and avoid that turning into a variation in image brightness. And the way we do that is to plot the images rather differently. Rather than plotting images as black and white variations in brightness, we plot the variations of the decay constants of the tissue. These are unaffected by sensitivity variations, and this process is called T2 mapping. Now, of course, to use an internal coil, we need to get it into the human body somehow and mount it on an instrument that clinicians will normally use for working on the biliary ductal system. And these are typically duodenoscopes. These reach down through the throat, right down to the opening into the liver, and they image nearby tissue. Or they're catheters. These are passed through duodenoscopes into the ducts that enter into the liver and drain fluid from it. So those are standard clinical tools. And what we've developed are thin film coils. These are little printed circuits that are very flexible. They can be attached to duodenoscopes or catheters, and we can use the same instruments for imaging as well as doing the normal tasks that clinicians use them for. Now, these coil systems must be intrinsically safe, and the other instruments, the duodenoscope and the catheter, they must be safe as well. And that usually means we need to make them out of non-magnetic materials so they can go in the scanner magnet, and they need to avoid being heated up by the radio frequency fields that the scanner generates. We've done a lot of work on this. We've developed non-magnetic tools, and we've developed little printed circuits that are not affected by the excitation signals that might otherwise heat them up. The way this is done is by segmenting the coils into little loops and twisting them. And as a result, the images that they generate, their sensitivity patterns, are quite complicated. You can see that they're segmented in the same way as the coils, and the image is only bright for a fairly small distance near to the coil itself. That's in the MR image on the right-hand side. Now, we haven't used these coil systems on living patients yet. Their safety is not proven yet. What we're doing instead is working with resection specimens from Thai patients at Concord University Hospital. These patients normally have cholangiocarcinoma induced by liver fluke disease, and they have undergone a resection operation to remove the cancer. And we can look at the specimen either immediately after the operation has taken place or a little time later. So here's an example. What we are doing is imaging a slice of tissue that's been taken from a patient. I have to say all these patients have recovered successfully and they have a good prognosis. And what is happening in the left-hand photos, you can see the duodenoscope receiver has been placed on top of a slice of tissue. It's being compared with the performance of a standard external coil, a torso coil that might wrap around the body, and we can have a look at the images obtained from each coil and see which one is better. So let's start with the external coil. That generates a standard MRI image. That's black and white. That's the image that's second in from the left. You can see the whole of the specimen there with some features on it that may mean something. It's a little bit difficult to say. There's some contrast, but maybe not too much, and you can see the whole of the specimen. If we now look at the T2 map, that's the colored image in the center from the duodenoscope receiver. You can firstly see we can't see the whole of the slice of tissue. The image is segmented. That's for the reasons that I previously gave due to the construction of the receiver. But now we see this colored map with some really quite clear features in it. We can see blue, that's the liver tissue itself. And we can see these green and yellow features. Some of them may even be brown, that are something else. So what are they? Well, this particular slice of tissue has been through the pathology lab, and the pathologists have identified those different features. I've ringed them in white and red. Some of them are inflammation caused by fluke eggs. The liver flukes lay eggs inside the liver tissue, and the tissue responds by kind of scarring around it. That's kind of a possible precursor to disease. It may or may not be. They're not so important, but the red ones, those are cholangiocarcinoma itself. And well, we're starting to be able to see all of these features quite clearly with these T2 maps, and I think in a much more immediate way than the rather dull gray looking image generated by the external coil. It's all pretty clear. Okay, now let's try something that's much more realistic. Normally, cholangiocarcinoma is a disease that grows along or around the ducts inside the liver. In this experiment, we've got a fresh specimen from a patient that's been resected possibly half an hour to an hour before. We're placing a catheter receiver inside the duct, that's shown in the photos on the left, and then the specimen is being scanned. We've acquired the conventional MR image using the catheter receiver. That's the black and white photo second in from the left. Again, that's fairly featureless, very hard to see what's going on. But look at the colored T2 map generated by the catheter receiver. Immediately, we can see what's going on. We can see the tumor itself as a brown feature that's got quite a long time constant, and surrounding it are gray and blue areas that are either liver tissue or fibrotic liver tissue. The brown is the important tumor. And even though the catheter receiver can't see that far, the image is quite restricted, we can see the important detail, namely the tumor around the ductal system. And over on the right, just to confirm, we've got the pathology data showing that there is indeed a tumor there. So how important is this likely to be? Well, what I'm showing here is a whole set of these T2 maps generated by that one catheter receiver in different parallel slices. You can see that the tumor itself isn't present in every slice. It isn't present in slice 9, but it is present in nearly all of the other ones. If you stack these up, which is what I've done in the three-dimensional picture on the right, you can now see the exact size and shape of the tumor. It's the brown feature that's coming in from the lower left of the catheter track, which is the same as the duct itself, and it's gradually encroaching on the duct. If we can generate maps like this, and if these are accurate, we may have the potential to know precisely which ducts are affected by tumor in the future, and also how to plan to remove them. So there is quite a potential here to improve the planning of resection surgery for cholangiocarcinoma. Not only that, the images have such high resolution that you can now for the first time see what is happening to the duct. It's got a lot bigger and thicker because extra tissue has grown. It's also become fairly irregular, it's not smooth, and we can see changes to the duct walls. So this kind of imaging may allow new scientific information in the future. So that's the basic idea. Let me summarize and give the next steps. We believe that these internal coils produce much clearer images in the immediate vicinity of the important part of the liver, the duct. They need to be introduced safely into the body. We've done a lot of work on this, and we think that what we've got is potentially fairly safe. And we've got a method of making use of the information to produce these nice clear displays in the form of T2 maps. And the lab experiments that we've done so far all look pretty promising, really. So the next steps would obviously be to try to image living patients. We have to be extremely careful about this. Obviously, we need to do further tests on safety. We need to check that there is no heating. We need to check that the patient won't be harmed in any way at all by this kind of process. And even if those checks are satisfactory, we need to make sure that the process will be of value. So for example, we need to work out what to do in the case where we can't insert these catheters properly because the tumor has grown and the ducts are blocked. We need to check whether we can still obtain good images even though the patient is breathing, because obviously that's a key difference between a lab experiment and a proper clinical demonstration. But if we can do this, there may be some potential for improving imaging, especially for surgical planning. So that's what we've done so far. Obviously, the pandemic has stopped us, but hopefully we'll pick up again when this is brought under control around the world. So for the time being, many thanks indeed to our sponsors, which obviously include AMMF. Thank you for attention.